Thank you all for being here to the third session of our virtual brown bag series on using R for agricultural data. We started out the first week with Kay giving us an introduction into using R for generating tidy data, tidying data sets, and organizing data in a way that facilitates analysis. Last week, Clark touched on doing data transformations and cleaning up messy data and some of the tools that R has for being able to manipulate data sets, combine them, split them apart, manipulate them in various ways to prepare them for doing analysis. And this week, I'm going to talk about how to do some more formal types of analysis. And so who am I? I'm Phil Alderman. I'm in the Plant and Soil Sciences Department, for those of you who don't know. I've worked a lot with R over the years, and what I want to do is to give you guys a window into some possibilities for how you might be able to use R to solve different computational or analysis type problems that you're dealing with your data. So what we're going to do for the session today, there's three main components. We're going to talk about how to use R for sort of generating summary statistics. We're going to talk about how to use R for analyzing designed experiments, data that comes from designed experiments. We're going to use a couple of different data sets for that. And then at the end, our goal is to touch on a little bit of some of the tools that are available within R to do exploratory data analysis and how we can use some of the automation and tools available in R to scale up our analysis and deal with fairly sizable data sets. So that's where we're going today. So I'm starting out here with a clean RStudio session. No project open yet, so I'm going to create a new project, as I do anytime I start an analysis. And I'm going to start that in a new directory, a new project, and I'm just going to put it in my active directory, and I'm just going to call this ag data analysis. Right, we're going to create the project. It's going to switch into the project. So now I have a project directory that I can work from. One of my first steps is to create a directory within that for storing all my R code. And then the next thing I do is to create a data directory, because presumably if we're doing data analysis, we usually have some sort of data file or something that we're working with. And being able to keep these in separate directories helps you keep things organized. You can find what you need and switch things out as needed. So the first let's see here. Let's get this started here. We'll call this demo, since we're going to demonstrate things, and we're going to save it in the R directory. All right, so as I said, we're going to first talk about some summary statistics and how that can be used. We're going to talk about analyzing designed experiment data, and then we're going to talk about using R for exploratory data analysis. All right, so those are our three goals for today. So I'm going to start out by loading the Tidyverse package. We've seen this before. It's really a meta package. It loads several different packages that are useful for being able to manipulate data in R. And then for our data, the first data set that we're going to work with for this demonstration is a data set that Clark was cleaning up, that Kay introduced in the first session, and that Clark did some cleaning on, and he produced an output file that had that cleaned up data for us. And so what I'm going to do is to navigate over to where I've saved that on my computer, and I'm going to copy it over to my current ag data analysis data directory. So I'm going to save that there, and then if I go back to my working directory, back over here to my project, and I look in my data folder, I now have that file there. So now I have some data in my data directory. I can now pull that in and begin to work with it. So it's a CSV file, comma separated value format. And so we're gonna read that in here. Okay, and so as you read it in, it actually gives you a little bit of summary about some of the as it's parsing things, how it's processed it. One of the really great things or important steps, I guess you could say, for doing analysis is first making sure that the data that you're going to work with for your analysis doesn't have errors in it, doesn't have mistakes or outliers or things like that. You have to make sure you're working with a clean data set. And thankfully, summary statistics are a really good way to be able to begin to explore some of that. And so R provides this function called summary, and it's designed to give you a summary of your data set. And so if I pipe we talked about the pipe last time, so if I pipe this weather data object into this function summary, and I run that, and then I expand this up so you can actually see, you'll see that it generates an output for each of the variables that's in my data set. And so I look here and I see, okay, well, I've got the date, which is actually the day of the month. I've got our temperature maximum, temperature minimum, and it gives you a summary of those variables. And so, for example, when we look at the minimum temperature, that's a numerical value. And so the way that it summarizes it is that it generates a summary statistics. So minimum and then the maximum gives you the mean or median, and then the first quantile and third quantile. So it gives you some idea of what the distribution of that column is, the data that's in that column. Likewise for temperature range. And so, as we look at this, it gives us a nice sort of big picture view of the data set that allows us to see, are there outliers? So for example, in the temperature range column, that's meant to be the range in temperature within a given day. So it's meant to be the difference between the maximum temperature and the minimum temperature. And as we look at the summary on that one, okay, minimum, sure, that seems about right. Mean, okay, that seems believable. Maximum, 323. Now, I guarantee you that there's nowhere on Earth where a 323-degree Fahrenheit difference within a given day, that's not a likely value. And so in all likelihood, that there's some error there, right? And so what we could do is sort and find that row and inspect further, try to figure out what happened. In all likelihood, and in fact, in this case, I'm pretty sure that what happened is that someone accidentally typed an additional 3 into that column. It should have been 32, and instead someone accidentally typed an additional 3, so it ended up being 323 when they were doing the data entry on this. So imagine if we were calculating, doing some sort of analysis with these data, and we had left, this outlier in. Obviously, that could have a huge impact. I mean, that's an order of magnitude higher than the rest of the values in the data set. And so, again, it adds an extra noise due to the error in that, then transcribing the data into the spreadsheet. And so, this gives us a chance to say, oh, okay, let's go back and figure out where that is, clean that up, fix that value, and we can move on. The other thing that we notice is that the temperature maximum here, this temperature maximum is meant to be a numerical variable. And so for some reason, it's not being read in that way. And so what we can do is to go back to this step where we were reading in the data, and we can add in a mutate statement, which basically modifies the columns that are in a data set. And we can say as numeric. We're going to convert that column into a numerical value. All right, and as we run that, you'll see that we actually get a warning message. It says that as it did that, NAs were introduced by coercion. What that means is that there was something in this column that could not be converted into a numerical format. And so as we then go to the summary, we see, if we scroll back up to that first column, we can see that, yes, there was one row where it couldn't convert that into a numerical value. Now, that's a problem on this column because it's supposed to be all numerical values. If there's something there that can't be converted, that means that there must have been some symbol in there or something that wasn't supposed to be there. And in fact, I did some checking on this beforehand. And what it is that the transcriber, the one who was doing the data entry, wasn't sure about the number that was in that column. And so they put a question mark next to the data that they entered. And so as a result, that question mark being there in the same cell with the number meant that the computer couldn't tell what number, how do I interpret this? Because it's got some extra stuff in there besides numbers. And so what it did was to just mark that as missing in the data. What we could do, though, if we really needed to, is to add some steps to filter that out, to clean it up, correct that, remove the question mark, and then convert it into numeric, and we would be good to go. In any case, I don't want to get bogged down in all those data cleaning kind of steps because we kind of covered some of those tools in the last session. Clark did anyway. So what I want to do is just to show so that you have the ability to summarize numerical data. It tells you how many missing values that there are in a column. And then what I want to do is to demonstrate also what it will look like for factor levels. So people that deal with qualitative data or categorical data, it looks a little bit different, and none of these in here have been converted into that. So what I want to do is to convert one of these columns into a factor. And so I'm going to do that for the wind direction, for example. So wind direction to add, factor wind EIR. So when we do this, what it's going to do with that column is that for each unique value of letters that's in that column, it's going to assign a factor level to it. And so when we then look at the summary of that, previously it had something like this, because character values don't have any readily, it's not easy to summarize character strings. And so, but once you convert it into a factor, what it'll do is it'll say, okay, these are our unique levels. So SE, NW, S, NE, SW. Okay, so we have all of these different here. And then what it'll do in summarizing factor levels is to give you a count for how many levels there are of that factor. Where this is really useful is when you're working with data sets that you expect that all of those levels should be the same. And maybe you end up with one that is the numbers of those factor levels is not the same. And so it allows you to see, oh, did some data entry not get entered properly? So sometimes, you'll get a space in here introduced into your factor levels by accident. And what will happen is it'll get separated out into two different levels of factor, one with a space and one without a space. And so what you can do is to use this to look for those types of patterns as you're inspecting your data set. As you do some of this data summary, it can be really, really helpful in identifying outliers and things like that. And we just talked about this one function. Gives you a lot of power even with that. Beyond that, let's suppose that you have a study and you're wanting to compare across years or across months or something like that. And in this case, we have some temperature variables that we want to summarize across years and months. What we can do is to summarize minimum temperature And what we do is we start with our weather data object. We can group by our month and year, okay, because we want to be able to have unique summary values for each month and year. And then what we can do is to use the summarize. They have both British and American spellings of the words. So there you go, depending on where your country of origin is, you can feel confident. Either spelling is fine. You can summarize the data set. And so what we can do is, for example, so we have in our data set this temp minimum for the minimum daily temperature. So it's the minimum temperature for a given day. And let's suppose we want to average it for these different months and years. And so what we could do is to do temp, or say tmin average is equal to, and then we're going to use the mean function to calculate the mean, and it's going to be temp minimum. All right. We can run that, and you can see that the output of that is basically a summary where we have each year, each month, and then our minimum temperature, average minimum temperature for those that particular month-year combination. So that grouping basically says that take all of the unique levels in month, all the unique levels in year, and for each unique combination of those, calculate the average of those values. And so this can be really useful, particularly when let's suppose you're wanting to create some sort of summary table for your publication, and you want to summarize what the weather was like, over the two different seasons of your trial or whatever. You can use this to generate that summary very quickly. The other thing is that sometimes you want to do this for multiple variables. And so let's suppose that we wanted to do this for the T-min and the we'll do it for the T-min and the T-maximum. And so one way to do that would be to spell all those out individually, but there's actually a capability here using the across function. And what we can do is to give across a list of columns. So temp, maximum, temp, minimum. And then we can then give it the function that we want to use for the summary. And if we only give it one, then it will generate an average here. And we had a missing value in that temp maximum, you remember from when we were talking about before. So actually we can add an na.rm here to remove that missing value before we actually calculate our statistics. And that'll clean that up so that we can get the average value for the maximum daily temperature and the minimum daily temperature for each of those combinations. And maybe we want to instead group it by year first. And if we change the order, it groups the results first ordered by the year and then by the month. Okay, so that's just one statistic for two different variables, but we can actually do better than that. We can actually provide a list of variables that we want. And let's say that we want to have the average here, and we want the minimum and the maximum. All right, we give it a list of the different variables that we want it to use. We give it a function that it's going to use to calculate those. And now within a few lines of code, I've generated a lot of summary values, average, minimum, and maximum for Tmax, for the maximum temperature, as well as the average, minimum, and maximum. It's not printed on the screen because it ran out of space, but let's see if I can expand this up. Let me see. There we go. If I expand that out and do this again, you can see that it's done that full calculation for the whole deal, both variables and all three statistics. So anyway, there's a lot of capability here for generating tons of different summary statistics. You could calculate the standard deviation, all kinds of different things in here, mix and match. There's a lot of flexibility in it being a statistical programming language. Unlike other programs like Excel or things like that, where you're having to point and click a lot, you can do quite a bit here in summarizing data fairly quickly. And so, and that summary can be really useful for getting a handle on your data set and getting a feel for what's there. And so, let's see here. Let me get back to here so we can see our data set. So, being able to generate the summary statistics is a very excellent first step in being able to get a handle on your data set. But typically, we want to do a little bit more than just summarize a data set. We want to actually get into generating some relationships, running statistical tests, formal tests for significance and things like that. And so that's particularly the case when we talk about working with designed experiments. So we've generated some hypotheses. We've designed an experiment to test those hypotheses. And then now we've collected some data and we want to actually perform those tests. And so once we've done looked at some of the summary statistics to check things and make sure everything's okay, the next step often that we want to do is to move on to actually analyzing the data from those designed experiments. And so to demonstrate this one, I'm going to use a couple of data sets that are provided in an R package called agridat. The reason I'm doing that is that those are already cleaned and have already been documented very well. And so this allows us to work with this data set. And I'm going to deal with a data set that's on some nitrogen application to some different varieties. And so let me go ahead and pull that in. That's going to be Yates oats. All right. And that's going to be, let's just do summary on that so we know what we're working with. Okay. And so we do summary. The same thing I did before. You can either put it on the same line or put it on separate lines. Like that. It gives you the same output over here. In any case, the row and column, that's some information about the design of the layout of the experiment, which row it was in versus column. The yield is the variable of interest that they measured. They have nitrogen, which is the amount of nitrogen manure that was applied to the plots. And then this GEN is for genotype. It's basically different varieties of oats. So we had different breeding varieties that have been released. And what they were interested in looking at was how do those different genotypes, or those different varieties, how do they respond to different nitrogen levels? Is there a differential response? Is it simply the genotype plus the nitrogen effect, or are there interactions between them? And so those were the questions that they were trying to answer. And so then we have a block column here in our data set, which indicates which subset of the field was used for applying treatments to. And so they had 6 blocks. And then within each of those blocks, then they had a split plot design. So the main plot was the genotype, and then the subplot was the nitrogen level. So combinations of nitrogen and genotype within these blocks, those are the treatments that we're interested in. So how does nitrogen level and genotype affect yield in this particular trial? And so so that gives us an idea of the data set that we're working with. We now want to actually perform some tests. We want to fit a model for our data set. And so to do that, we're going to use a type of model called a linear mixed effects regression model. And that's LMER. And to do that, we actually need to load a couple of packages that are going to help us with that. So I'm going to scroll back up to the top of my file and add, let's see, library, I can spell. LME4, that's going to give us the package for, or the function for fitting the model, and then I want to perform some tests with it. And so we're going to load both of those packages. And now we're going to scroll back down where I was before. Okay, so now we want to work with this oats data set. Now, because there are so few levels for the nitrogen, I want to treat that as a factor. So instead of treating it as a continuous variable, I'm going to treat it as a categorical variable, that each of those different nitrogen levels, because there are so few of them, I'm just going to treat them as a factor. And so in order to do that, I'm going to modify that column. In fact, what I'm going to do is create a new column which contains the factor level. So mutate in fact for nitrogen factor, and I'm going to create a factor out of the nitro column, which has the different nitrogen levels in it. Voila. Okay. So now we have a data set that has both the nitrogen column, but now we have a factor level. You can see that the values are the same, but the internal representation of these is different, and will allow us to analyze the data in a particular way. So what we want to do is to first determine whether or not interactions are present. And so what we're going to do is first fit a model with only main effects. So fit model with main effects. All right. So to fit that, we're going to say yield as a function of our in factor plus our genotype. And we're not going to put in any interaction term between those at first. Right, and then what we're going to do, so this is a mixed effects regression model. So we're not only going to look at fixed effects, we're also going to look at random effects. So, and for that, we're going to fit a random intercept for the block, and actually it's the genotype within the block. Because this is a split plot design, it's not just the block, it's the genotype is part of that nesting structure. So, all right, so we have the block effect and then the genotype within the block as the random effect. All right, so that's what those are the components that we expect to be randomly varying. We're not interested in the specific levels of the block. We're interested only in the overall effect of our factors of interest, which is the nitrogen and the genotype. Okay. And then for our data, we want to use the oats data because we had added that column that treats nitrogen as a factor. All right, so what we can do is fit our model, and then the model fit is stored in this oats.main object. And what we can do next then is to fit a corresponding model, but include interaction effects. And to do that, we can add it directly this way, in fact, :GN. But there's actually a shorthand notation for this, and that is to simply change the plus to an asterisk. And that that's a shorthand for each of the main effects plus the interaction between them. All right. So if we do that, oops, we overwrote to rename it here. Fit our main effects, and then we fit our interaction effects. And then the function that you use to compare them is a bit funny because it's not actually an analysis of variance, but you use that function to do it. And so it's oats main, and then we're going to compare that to oats with the interaction effect. And when we do that, you can see that it is performing a likelihood ratio test. And the comparison of the likelihood ratio, the likelihood ratio test, the output of that basically tells us that there's not really any significant difference between the model without the interaction versus the model with the interaction. So on that basis, typically we tend to go with the model that's simpler. And so for our subsequent inference, then we would proceed with this oats.main. And we could test all kinds of things. We could say, was the genotype effect significant or not? Or was we could mix and match with the fixed effects to fit simpler models and then do that comparison here to determine which is the model the simplest model that still performs just as well as the more complex one. So anyway, for our purposes here, we can then proceed with this analysis with looking at the true type 3 Anova. And so what this does is to generate a type 3 analysis of variance table. And with mixed effects models, it's a little bit tricky because the denominator degrees of freedom is not straightforward to calculate. And so it tells you here that it's using the Satterthwaite's approximation for calculating the denominator degrees of freedom for being able to determine the significance level for these different factors. And so we can see here that the nitrogen appears to be significant, and that there's not much evidence to support the genotype having a significant effect. So typically, we use a value of 0.05 for our probability, and so we would then conclude that the nitrogen factor is significant and that the genotype effect wasn't, at least in this particular study. And so if we are interested in the particular effects, we could actually summarize that model object, and it will give us the actual estimates for those different factor levels here. And so it gives us information about the random effects that were fitted, as well as the fixed effects. So anyhow, that's one data set. I want to touch on another data set, which is a little bit different, but still a designed experiment from an animal science data set. So any questions about that so far? I know I've kind of been blowing through things here. Okay, well, I don't hear anyone speaking up. Please speak up if you have any questions. I'm happy to address those as we go, or drop them into the chat, and we'll try to answer those at some point. Okay, so the next data set that I want to demonstrate here is a data set with calves actually. And what I'm going to do is to summarize that data set. So this data set is also coming from that Agridat package. All right. And so we can summarize it to get a feel for what's going on in that data set. So for this study, what they wanted to do was to look at the effects of iron supplementation and a particular pathogen that tends to affect iron uptake. And so what they had was a 2x2 factorial experiment where they had treatments where they either provided iron supplement or did not provide iron supplement. And then they then were looking at whether the animal was infected or not. So whether it was the inoculation of that or not. So looking at the interaction of those effects was their target. So was there an interaction? Is it simply that, one or the other affected things? And so, but for animals, particularly young animals, they're growing fairly rapidly. And so what they were interested in is how does this affect their growth rate over time? And so they had all of these animals, they took multiple measurements on each of the animals. And so we have essentially what you would expect with this repeated measures is that there's probably some sort of inherent growth potential for that animal. And then on top of that, these treatments are having some kind of an effect. And so what we want to do is to say, okay, well, we don't care about necessarily the predictions about those specific animals. What we want to do is to be able to predict for animals in general, but we want to be able to account for the proportion of the variance that's affected by whatever that growth potential of that particular animal is. And so what we can do is to use a similar type of modeling approach with random effects, with the animal being our blocking factor. So for that, we're going to look at the animal weight as our variable of interest. And I've neglected. There's actually a couple things we need to clean up for this data set. And let me pull up my notes to make sure I do that right. Okay, so we see here that we've got a missing observation in weight. Sometimes that can cause problems for us. So it's actually better to go ahead and clean that up. So let's go ahead and do that. And we're going to call that clean data. What we're going to do is to filter out that missing data. So we're going to say not is NA. So it's not missing. We're going to keep all the ones that are not missing and drop the one that is missing. And then we're going to set our reference levels for our factors so that when we do our model fitting, we can interpret the output in terms of so the control treatment here is that there's no iron supplement and they're not infected. And so we want that to be our baseline. And so we want to re-level the factors so that our baseline is this no iron. So what we're going to do is to say iron factor re-level iron, and then we're going to say no iron is our base level. And then we're going to say effect equal to factor level. Non-infected, right? And so by doing that, we now have established that our baseline is that control treatment. And then I'm going to just redo the time variable here. I have to rescale it. Sometimes that makes things a little bit easier for us. So basically, the first day that we have data for is day 122. That's when they initially weighed them before imposing treatments. And then we're dividing by 10 just to kind of put things in a easier to work with scale. So, all right, so that's all the transformation that we need to do, I think. Now we can actually proceed to, let's see. And this is going to be weight. And we know that there's a time trend because these are our calves that are growing. So we're going to put in, not day, but time rather. And then we're going to add our iron treatment here and our infection, whether they were infected with pathogens. So those are our fixed effects. And then we're also going to look at then a random effect for the animal. All right. There are a lot more complex ways that we could handle this, and it's a repeated measure. So actually, there's a more complex variance structure we could explore, looking at autocorrelation or something like that between different dates. That's all possible. I'm not going to get into that here just because I don't want to bog us down too much. In any case, we could look here and pull out the clean data, and then, all right, so then we fit our model for the main effects, fit model for main effects, and that's basically just iron plus infection. We assume that there's no interaction for this model, right? And then we likewise will have one with interaction. And for that, as I noted above, we can just add an asterisk. Oops, not an 8, but an asterisk. And we're going to change this to infection. And we can run that and run that. And now we can compare those two models just as we did before. So compare models. All right. Just as the previous model, we find that there was no significant interaction, and so we'll proceed with the main effects only model for inference. And for that, we would just do our Anova main. And for that, we'd see that, okay, both the iron and the infection of the pathogen were significantly affected growth, as did time, but that's kind of a no-brainer. And so then comes the question, okay, well, what kind of an effect was it? Was it a positive effect, negative effect, et cetera? And for that, we could use our summary to get more information about that model to determine that, okay, both the iron and the infection were negative effects. And so, with certain probability associated with that estimate. So we can, so that's two different data sets with designed experiments. As I mentioned, this is a repeated measures experiment. So typically you would want to at least test to see for homogeneity of variance across the different measurement dates. And so you would fit several models probably with different variance structures and inspect them to see which ones are a better fit for that variance, and then proceed with inference based on that. In any case, the goal here is just to give you an idea of the range of things that's possible within R. So this would be a very typical kind of way of analyzing data. A lot of people analyze their data this way using SAS, for example. And so you can do all of that and more here. Nothing against SAS, but anyway, just to demonstrate the potential for R here. Okay, so for our last little section here, I want to touch on the data set that Clark touched on last time. And this was the one of country crop yields. And I'm going to copy over. So he did some of the cleaning up of things. And so I'm going to copy that code over here, just so we don't get bogged down in talking through all this before. Basically, reading data in, extracting out the top countries in terms of population, and then tidying up the crop yields, renaming some things is what's going on. And then he finished up with generating this fancy schmancy plot here, showing the crop yields for different crops over time. And so that's, looks nice and lovely. But maybe we have some different questions about what's the rate of change per year for these different crops, and how might we be able to use some modeling tools to be able to access that information and do something with it, okay? And so, we can see, maybe there are some that are going up, most of the yields are going up over time, but maybe some are declining, and we might have some questions about which ones are increasing, which ones are decreasing, et cetera, et cetera. And so we could kind of eyeball it on some of them, but maybe there's a better way than trying to do that visually. And so to do that, what we can do is to use a package, which is actually a meta package called Tidy Models. And so we're going to scroll up here to the top and add I prefer to keep all my package names at the top of the file. That way I can tell what the dependencies are every time I'm opening the file. Anyway, so Tidy Models, I'm going to load that one. And you can see that it in turn is loading some other packages here that are really useful for working with different model objects of different types. All right, so if we scroll back down here, we've got this tidy data now with crop yields over time for the top 30 most populous countries. And what we can do then is to do some modeling with it. So if we start out with our tidy yields data set, what we first need to do is to prepare our data for this process. So let's suppose that what we want to do is to look at the slope, in other words, the rate of change over time in yields for these different countries. Okay, so what we want to do is to fit a line for each of these crops within each of these countries. And so to do that, we need to actually package the data, repackage the data so that we can do that. And so what we're going to do is to use this function called nest, which allows us to select some columns from within our data set and pack those down. And so what we're going to do is to say we want to grab the year and the yield. Okay. And so what that's going to give us, I'll just run it like that. And actually, let me just do it this way, the view, so that you can see the output there. All right, so we've previously, up above here, we've grouped our data set according to the different countries and crops and so forth. And so when we nest it, it nests within those groupings. And so we now have for each country and crop, we have essentially condensed down the data on yield, year, and yield into its own little sort of mini data set within this column. And so each row of this column is actually a data set within itself. And the reason why that's useful is that we can then use that combined with some linear modeling to fit for each of those little mini data sets. All right, so what we can do is continue our pipeline here, and we're going to add a new column, model, and we're going to use this function called map, which basically just goes over each element inside that column. And the one we're going to do that for is our nested column yields. All right, and what we're going to do for each of those mini data sets is to fit a linear model for yield with respect to the year. Right, and our data equals .x. Okay, so this .x basically is referencing one element out of this whole column. So you remember before we had this column that, let's see, as we did, let me just add this. I shouldn't have closed this before. Okay, so each one of these is represented by this .x. So basically what we're going to do is to fit a model for each row of that data set. All right. And then rather than dumping it to the screen, we're going to assign this to IDLM. Oops, forgot to take off this little thing here. All right, so now what we have, if we want to view it, view IDLM, is our original column here, which was the yields data packaged up. And now you can see we have a new column in here, model, which contains, if you kind of mouse over that, you can kind of see that it's got different model fits stored within it. So each model fit, there's information about the coefficients and things from that model fit. And that fitted object for this data set is stored in this element here. For this data set, it's stored here, and so on and so forth for each of those little mini data sets that we created. All right, and so we can then extract the slopes from those, because what we said we were interested in was what is the rate of change over time of yield for those crops in those countries. And so what we can then do is take our data set with that column of fitted models, and we can pipe that in here, and we can use a function here So this model column is the one column that we created in the previous step up here. And the function tidy here comes from the yardstick package, and it basically has a way of tidying the model output and creating something that can be packaged into our kibble. And so as an output of that process, we actually have, it's a nested object. And what we can do is to unnest that object, which will basically expand out all of the coefficients that have been extracted from that object. And so we'll unnest that. But then the one that we're interested in are the coefficients for the year term. And so we're going to then filter that based on the term equals year. All right. And so we're going to assign that slope that way. Oops. You have to type it. Can't misspell it or it doesn't know what you're doing. There we go. So now we have this slopes object that we can look at. And you can see that it's got, okay, there we go. All right, so we've got the country code, crop, our mini data set, our model column, and then we now have a bunch of other things related to the year. So for the year, we have this year term. We've got the estimate. This is the estimate of the coefficient for the year term that are from all of our linear models that we fit. And then the units on that are going to be in the units that we fed in. So I believe the yield was in tons per hectare, and then per year. So this is the rate of change, how much increase or decrease in the tons per hectare per year is what this estimate of that slope would be. And so we have both the estimate and then the standard error of that estimate and the t-statistic, I think it is, as well as a p-value associated with that, keeping in mind that p-value doesn't account for multiple comparisons or anything like that. But anyway, what we're mainly interested in here is the estimate, just for exploratory purposes. And so what we can do is to then take that slopes data set, feed that into a filtering statement. So let's say that we're interested in looking at the increase in wheat yields over time. Okay, so we could say crop equal to wheat. And if we do that, we see that we can filter down to now all we have here are estimates that are related to the wheat. And then we can further do that and say, okay, let's say that we want to arrange, we want to sort them all in rank order from the greatest gain in yields to the least gain in yields. And so what we can do to do that is to use this arrange function, descending, so from largest to smallest, and then use that estimate column to do that ranking. And if we do that, we can see that Germany had the greatest yield gain over the period of the data set, followed by Egypt, China, UK, so on and so forth. So obviously, this is one particular set of things that you could look at for a particular analysis. It's a large data set. Sky's the limit in terms of the different dimensions you could go in exploring this data set, combining it with other data sets. But this gives you an idea of the range of possibilities. This is a fairly sizable data set. It would take some work to kind of manipulate all those and get them in. But using some of these really handy tools in R, it allows you to take on quite a bit and do some pretty cool work. So in any case, let me leave it there. If you want to see a visualization of this output, then you can come back next week, and Kay will be able to demonstrate a visualization of some of this model output, as well as a visualization for some of the other data sets. And for that, we'll talk a little bit more about this tool up here that ggplot that Clark used and I used a few minutes ago. But anyway, hopefully that gives you an idea both how to use R for summary analysis and summary statistics, checking things, doing analysis for a more designed experiment, or if even exploring fairly sizable data sets and looking for patterns and things like that, as we did with that final data set. Lots of capability. I encourage you to take advantage in the future with some of the different resources available at the library of training and some of those kind of things to give you the more hands-on experiential learning of how to work with these tools. There's a lot of power there, so hopefully you'll take advantage of those opportunities.